Okay, well, thanks, Sam, for the introduction. So my talk today will focus, I think I probably put the most general title to make sure that I can discuss as many little aspects that I've been studying throughout my PhD, my first postdoc, and now that I'm integrating in my current research. But I do work on multiple stressors, tipping points, but at the effect level and how multiple stressors impact individual organisms. I've been focusing on model species that are way less sexy than the species that Brad showed you. So I unfortunately do not have cute videos of giant squids feeding, but great model species for what I'm doing. So I'm not gonna, we all know that we have a lot of ongoing changes in marine environments with global change, stressors such as ocean acidification, global warming, hypoxia. But we also have a lot of local activities that create very local stressors, such as pollution and contaminants. We have a lot of emerging contaminants that we don't know a lot, such as the properties and effects, habitat destruction that can be very localized, and invasive species. So all of these stressors will, of course, overlap in time and space. And we have noticed that in coastal habitats where the biodiversity is quite elevated, there's also a convergence of stressor. So this is the work of one of my friends and collaborator, David, who looked at the cumulative exposure of multiple stressors in the St. Lawrence estuary. And we see that there is this convergence of stressors in highly populated areas and also in coastal areas, which is not such a big surprise because we have the convergence of stressors coming from the marine or the aquatic environment and the stressors coming from the terrestrial environment. But this means that we have a hotspot of biodiversity accumulated to a hotspot of stressors. And all of these stressors can interact. So this is a very schematic way of looking at different stressor interactions. So let's say that all of the little rounds are different stressors. We can have intensity interactions that occur when one stressor changes the intensity of another stressor. We can have an effect interaction that happens when one stressor changes the effect of a given intensity of another stressor. And we can have what we call high order interactions, which basically occur when the interaction between two stressors is altered by a third one. So all of these stressor interactions will have different effects on marine ecosystems. And they're going to have effects at different levels of biological complexity. So of course, they're going to impact organismal processes such as genetic, molecular, physiological, and behavioral impacts. They can also affect populations, so they can shift dynamic in the distribution or just the range and the pattern of different populations of species. And ultimately, it's going to impact whole communities, so species interactions, trophic transfer, the complexity of community structures, and eventually ecosystem processes such as nutrient cycling, carbon sequestration. So these are just examples, of course. But they will have impacts at different levels of biological complexity. Of course, we can see the effects of multiple stressors. So you've probably heard about synergistic and antagonistic interactions. So when multiple stressors are present at the same time and at the same place, they can create what we call synergistic effects. So if we look at this simplified figure, there's going to be a stressor A that has an effect compared to the control, so an impact of 5, let's say here. We're going to have stressor B, which has a different impact, and we're going to have a synergistic effect when the response that we observe when both stressors are combined are higher than the addition of the effects of those two stressors, and we're going to have an antagonistic effect when it's lower than what we would expect in an additive manner. So if we simplify, for synergistic effects, we have amplified effects, and for antagonistic effects, we have attenuated effects. And I think we've been trying to kind of create generalities in terms of the effects of multiple stressors across different biological levels, across individuals and communities and ecosystems. So this is work by Cote et al., which was published in 2016 when the concept of multiple stressor was really really took off. And we can see that there's no really generalities on what we can expect for synergies and antagonisms if we look across physiological, individual, population, community, and ecosystem responses. Because ultimately, what modulates the effects of multiple stressors is going to be the performance or the response of one organism to a given stressor. So this is just a performance curve and they can take different shapes, of course, along an intensity for one stressor. So of course, it can be altered. You can see an increase in performance or a lower performance, or you can also have a shift in performance depending on different factors. And I've kind of been interested at all the different factors that can impact this shift in performance in the context of multiple stressors. And I kind of realized that there is a lot of stressor of factors that we can take into consideration. So of course, there's environmental context. So what combined stressors are we looking at? What's the acclimation history of the individuals? What are the daily and seasonal cycles, the food availability at the moment, the sequence of exposure, the duration of exposure. So these are some examples. Of course, there's going to be some intrinsic biological factors, such as genetic variation, phenotypic plasticity, the life stage that we're looking at. So we've seen that the effect of single and multiple stressors really varies with the life cycle, the sex, body size, physiological state. So this is just to name a few. There's going to be evolutionary context and also experimental and methodological influences. And all of these can also interact to kind of modulate what we see at the effect level. So this was just my introduction to say that I've been considering a few of these factors in my research in my PhD and my first postdoc, and I'm now switching to a different approach that I'll be speaking of a little bit towards the end. So one of the first factor that I want to talk about is the stressor intensity and combination. So this is my third chapter, I don't remember, one of my PhD chapters where I investigated the effect of nutrient enrichment in the presence and absence of saltwater intrusion in the St. Lawrence. So we looked at the zebra mussel, which is an invasive species, but it was a great model species to look at because it's now everywhere in Quebec. It really took over, and we wanted to kind of know, okay, what explains its presence everywhere. Nutrient enrichment, because where we find it, we have a majority of the land around it is agricultural land, and we do have a problem with nutrient input in the water. And why intrusion of salt water is because in Quebec, we have the salinity front that is shifting towards the freshwater part of the St. Lawrence. So we have punctual arrivals of very high saltwater intrusion. So this is just mortality percentage along the nutrient enrichment that you see right here. In green, we have the stable salinity, and in yellow, we have the same gradient but with the presence of a weakly saltwater intrusion. And what we can see is that in the absence of salt water, we have this quite linear response up to a point, and then we see a threshold. So beyond a certain nutrient enrichment, doesn't matter how much you increase nutrients, the mortality kind of stays the same. So we identify a clear threshold, which corresponds to a tipping point with further analysis that we conducted. But when you add saltwater intrusion, we kind of have this weird pattern with two thresholds in our response. And we kind of see that nutrient enrichment can kind of have a positive effect in the presence of saltwater intrusion between those two levels. And then I was interested in looking at what type of effects do we have along the gradient. So in red, we have synergistic, so amplified effects. And in blue, we have antagonistic responses, so attenuated effects. So we do see this shift along the gradient in terms of the effects of both stressors, which is kind of interesting, but then becomes very kind of complicated because if we look at only one intensity, at one intensity for a stressor, then it doesn't mean that we're going to capture the whole effect when it's combined to a different one. Then I did another experiment. That was my fourth chapter where I looked at different time of exposure. So this was a very fun experiment that we did in situ. And we looked at nutrient input temperature on blue mussels, so as an engineering species, at different time of exposure in the marine estuary. So what we did is that we basically recreated our mussel transplants, and we had different abundances of mussels because I was also working with another student who was interested in the complexity of engineerings and how they modulate the community structure in the presence and absence of multiple stressors. So basically, we did our transplants and we exposed them to nutrients and temperature. For the temperature, we kind of recreated a heat wave at low tide using those kind of greenhouses that we made. And we did different thermal stress during the summer, and we looked at multiple responses at different time of exposures. So this is just one of the responses, and what's really fun is that I looked more at the individual level and on the blue mussel, but Felix, who I worked with, his paper is currently under review, really looked at community structure, the amount of sediment accumulated in the mussel patches, so really looked more at the community and ecosystem level. So we're also going to be linking those results to see what happens from the individual to the whole ecosystem processes. But this is energy content, so the energy using a calorimetry bomb. And we had a statistical interaction between thermal stress, nutrient, and time of exposure. So if we look at what happens exactly, we see the individual and combined effect of nutrient and temperature after exposure 1, but we see that after exposure 2, we have a net increase in energy content when both stressors are combined. Energy content, however, can be either a response to stress or it can be a positive thing. It could be also an increase in food availability at this time of the year because we were in a natural environment. So it's a little frustrating that we can't dig deeper into the interpretation of this result, but we do see a shift in the type, in the effect of multiple stressors across time. So this brings me to another experiment that I did recently in my first postdoc, where I looked at, I'm gonna have it after, where I looked at temporal dynamics in multiple stressors. So this is really a factor that is understudied in the context of multiple stressors, and I looked at carryover effects. Carryover effects have been, I think, defined in 1,000 different ways, but let's keep it simple. For me, in this presentation, is where past environmental experiences can influence an organism to future exposure of multiple stressors. So I worked with Paul Brooks, who developed a very cool in situ experimental system that can manipulate different doses of stressors and different combinations. So once again, that was on the blue mussel. And I looked at the pre-exposure of nanoplastics and how this affects the tolerance of the blue mussel to freshwater input, nutrient enrichment, and copper input. And just a small parenthesis on why nanoplastics. Well, I looked, as Sam said, my first postdoc was to characterize and understand the impact of nanoplastics in the context of multiple stressors in polar systems. I am not a chemist, so I will not go into the nanoplastic characterization aspect. I had the amazing job to go and sample in amazing places to bring the samples back, and we had an amazing team of chemists and that developed protocols to identify nanoplastics in different abiotic and biotic compartments. And not a surprise, we went to the North Pole and we even found some nanoplastics in the ice in the North Pole. So in keeping in mind that nanoplastics are everywhere, which is not a surprise, but we still needed to show the type of nanoplastics and what concentration. I came back to do this experiment in Dublin, looking at the blue mussel because we were working with indigenous communities and they had a particular interest in the blue mussel. So what we did is a pre-exposure to nanoplastics for 6 days, and then an exposure to multiple stressors, so nutrient enrichment, copper contamination, and freshwater input for 3 weeks. Why 6 days? It wasn't completely random. We first did an experiment where we wanted to look at the time of exposure and concentration of nanoplastics on energy content in the blue mussel. And we only saw an effect of the concentration after a 6 day of exposure. So if we have less energy in the mussel, we can expect that their tolerance to multiple stressors afterwards will be altered. And a lot of people will say that the impact of nanoplastics is probably not the plastic itself. And we tend to agree with this. We also, because during our experiments, we use what we call environmental nanoplastics instead of engineered nanoplastics, which have been used a lot in the past. But environmental nanoplastics also have all the contaminants associated to the plastic, instead of which engineered nanoplastics don't have. And we do see after 6 days of exposure that we have an increase in those contaminants also in the mussel. So we believe that the effect probably comes more from the metal or the other contaminants associated to the plastic than the plastic itself, because it's so small. It just goes through any biological barrier. So it's hard to characterize the impact of that very small particle. So based on this, we did the experiment. We saw the effect of individual stressors on different, on the survival. So we had an effect of nanoplastic pre-exposure overall, we had an effect of salinity, and we had an effect of copper on survival. But what was interesting and a little bit mind confusing is that we had a significant interaction, statistical interaction between copper, nutrient, and nanoplastic pre-exposure. And unfortunately, we have a lot of marginally significant results, which complexifies a little bit the interpretation. But what's really interesting, in the absence of nanoplastic, we have an effect of copper that kind of switches in the absence and presence of nutrients, that kind of switches when the nanoplastics is present. So it kind of seems that the nanoplastics pre-exposure changes the effect or the direction of effect of stressors of copper here in the presence and absence of nutrients. So it's a little confusing when we have three-way interactions like this to understand what is the main driver what is the main stressor that kind of drives the interaction. But we can also see this is HSG, so it's effect sizes, which is kind of another way of characterizing stressor interactions. So we see the effect of copper compared to control conditions, the effect of nutrients, the effect of nanoplastics, and when it's combined. So the combined effect is almost as high as the copper, but we do see this synergistic interaction between all three, this synergistic effect. So which means that the pre-exposure to a stressor can alter the effect of different stressors afterwards. Complexifies things a little bit. Now at Trinity, I've been focusing on a complete different species. I've been working on Daphnia magna, which is a very good model species in freshwater ecosystems. So this is the work of my first master's student, who I'm really proud of. Her work has been accepted, it's in press. She was interested in responses to multiple stressors within populations and across populations of Daphnia magna. So basically, she took 3 clones from France and 3 clones from Maine that are genetically different, but that are still part of the same populations. And she exposed them to copper, elevated salinity, and the combination of both. And what's really interesting, of course, we expected differences across populations. That was the hypothesis, but we didn't expect to see as strong differences within the same populations across different genotypes. So that was quite surprising. So this is the mean days alive, which is a proxy for survival. And we see already these intra-population differences for copper, but we do see that these differences are even more prominent in the context of multiple stressors. And if we look at the total number of offspring, which is a common performance or trait in Daphnia magna, we see that this intra-population differences is even more pronounced. So that was a really, really cool finding, which kind of made us want to investigate what happens at the genetic level when we have in the context of multiple stressors. So, as I said, I'm using Daphnia magna as a model organism. So, of course, it's a perfect indicator of change because it's quite sensitive to environmental stressors. It has a really, really quick generation time, which allows to do transgenerational experiments in the context of multiple stressors. And it has a I always struggle to say that word, parthenogenetic reproduction, which means basically that it always recreates clones, and it only creates female clones, and it will go into sexual reproduction only when it's stress. So we only work on females, which means we have an infinite number of clones, which is kind of really nice for controlled experiments for multiple stressors like this. So this is the current work of my master's student, Aisha, who's interested in the effect of salinity and copper on different genotypes in Daphnia magna. So she did a pilot study that turned out to be an experiment with 144 treatments. So I don't think we can really call it a pilot study at that point. But she looked at a gradient of 12 intensities for salinity and 12 intensities for copper, and she tested all possible combination. And she did this across 3 genotypes. So a Finnish genotype, a genotype from France, and one for you guys, one genotype from Belgium. What is really great is that you can have really short exposures in Daphnia magna, and you can look at survival, which is a proxy, which is a quick proxy to have these effects, to see those effects. And what we can see, so she literally just finished her second block of the experiment 3 days ago. So I analyzed the results very quickly and want to show this to you. So I'm sorry for the lack of interpretation, but we saw that although our genotype B was meant to be the most salt tolerant, because it came from rock pools in intertidal environments, but it turned out to be the most sensitive to both stressors, which was kind of a surprise for us. And for one of the genotypes, so for genotype C, copper toxicity depends on salinity. We saw some threshold effects for salinity for all the genotypes. Same thing with copper, but except for copper, what we saw is that the genotype C responds linearly to copper. So it means that basically different genotypes will respond differently, and that performance curve will be quite different according to the genetic background and the clone that you're using. And this is what I will be working in the future. So I have the amazing chance to be working with Dr. Ebert from Basel University and Pepijn Maux. They have developed the Daphnia magna GWAS panel. So they basically have a panel of 256 different genotypes of Daphnia magna from all over the world. You can see them here that have all been sequenced. So once again, this is amazing. And they are both, the panel is both present in Trinity and in Basel University. What I'll be doing is using that GWAS approach, which is genome-wide association study, which basically scans the whole genotype to attribute a trait or a phenotype to a SNP or a genetic region. So it kind of reveals a region or SNP, and it kind of reveals a mechanism that can link that genotype to a performance to eventually enable ecological predictions. So this is a work of a PhD student that I'm working with. She worked on a parasite in the context of multiple stressors. She just finished her thesis. I wasn't involved in her work here directly, but I've been working with her for the GWAS now. And she identified, so basically this is a Manhattan plot, and you can identify a gene, which we can see right here, or at least a region that is involved in the tolerance to one stressor, which is right here, a parasite. So this is kind of what it can give us in terms of information. And then you can understand, of course, what the role of that gene is afterwards. So what I'll be doing, and it's actually happening right now. So I have a few students working with me that are keeping this going as I'm away, having fun in Kristineberg. So I feel a little bit bad, but we need to keep this running. So what we're testing right now, based on Aisha's results that I just showed you, we're testing a gradient of salinity. That same gradient of salinity combined to one one intensity of copper, and then we're testing a copper of gradient and to copper combined to saltwater input. And we are testing this across 256 genotypes from the panel. So this is work that started 2 months ago, and I think we still have another 6 months of experiments. But the exposures, as I said, are very short. And what we'll be doing, depending on, because right now we're only looking at survival with this short exposure, but depending on the results, what this is going to allow us to do is to run bigger and longer experiments and look at more traits, but looking at specific intensities for copper and specific intensities for salt, which will allow us to dig even deeper in different mechanisms. So we're going to be looking at offspring production, the ratio of female and male that they produce, which is a direct response to stress, and also different metabolic and responses and physiological responses. Yeah, and eventually what we want to do is to identify genes that are involved in the resilience, tolerance to one stressor, the other stressor, and when both are combined, and to kind of understand what happens at the genetic level in the context of those two stressors. I'm going fast. So why am I here now? So I've been wanting to work with the Sam and Laurent for a while now. So one of my main objective was to come and learn how to work on acidification, which is a great setting to do here. What we're working on now, and we've decided on the design yesterday after doing many tests. We're going to look at the effect of pH on bioluminescence in this brittle star. So this is a very ugly drawing, my apologies, but it's just to show that this brittle star has those daily movements in the sediments and at the surface of the sediment, so it's exposed to different pH daily, but we want to know what the effect of different pH, so the natural variability and the natural exposure, and under future scenarios, how this affects the bioluminescence in the species. So right now we have some preliminary results. We're just starting the experiments now, and it's going to be going on until I leave, so until the 12th of November. And yeah, we have many more questions to after following this, but we're already seeing quite a nice pattern that we're excited to show you eventually when we have more results. So of course, all this work that I've showed you is from my master's, PhD, and my first postdoc. So it's not, I didn't do this alone. So I want to thank all these collaborators. And although I'm still here for a while, I still want to take the chance to thank Sam, Laurent for their help in this experiment. And thank you for being here. It's nice to have you, although I understand that it's a holiday for you guys today. So.
Thank you.